Hi everyone, welcome to this week's module. This week we're going to be focusing on biological molecules. Now we're going to start off looking at the inorganic molecule water. We're going to look at its structure and discuss how that structure relates to its function within the body. We're going to move on then to looking at macromolecules, which remember from last week are at that basic level of organization where we look at that chemical level, right? We take atoms, put atoms together to make molecules, and then take specific molecules and put them together to make our macromolecules. All right, so we're going to jump into that level, the chemical level, looking at macromolecules, how we build them up, how we break them down, and then we're going to focus in on the first two, proteins and nucleic acids. So all living things are composed of water. A large proportion of our body, about roughly 60%, usually about in an adult male, it's about 60, and a female, about 55% of the body is composed of water. And you can see here within the body, looking at all of these different organs, for the most part, roughly we're here about above 70% total composition of water in most of these structures within the body. Now, if you take a look here at our skeletal system, the bones, okay, bones are made of about 22% water, so definitely on the low end. Another tissue that's not listed here is our adipose tissue, right? Adipose tissue is our fatty tissue, and fats are hydrophobic, meaning they repel water. So adipose tissue also has very low water content. So total body water, as I mentioned, in an average healthy adult male, you're looking at about 60%, and in the adult female, about 55%. And one of the reasons there, I just actually alluded to, we were talking about adipose tissue and how it has a much lower total composition of water. In females, females have breast tissue, and breast tissue is primarily fatty tissue, and so that makes up that difference. So because there is a higher proportion of fatty tissue in the female body compared to males, females have roughly 55% of their body composition made of water, whereas adult males have about 60% of their total body composition made of water. With infants, infants are about 75. Okay, so I want you to take a second and try to think, why would an infant be significantly more? About 15 to 20% more total composition of water. And it comes down to something we had just talked about, bones. So with infants, their skeleton is still highly made of cartilage. Okay, so an infant has a cartilaginous skeleton. The skull bones have not yet fused. Ossification, so the process of turning that cartilaginous skeleton into bone, has started to take place, but is not fully occurred at this point. So they have a lot more cartilage in their body and less bone. Okay, so the decreased amount of bone means that they have more tissue that is higher in water. Okay, so that's why infants are roughly about 75% water, whereas when you become an adult, you're about 60 and 55. And then as we age and get older, our muscle tissue, which is high in water, becomes replaced by more fatty tissue. And so you also see a decrease in total water composition as we get older. Now, water is known as a polar molecule. Water is slightly charged, and there are two different charges here. You have slightly positive charges with the hydrogen ions, and then slightly negative charges on the oxygen. All right, remember, water is made up of two hydrogens and one oxygen, H2O. So you have slightly positive hydrogens and slightly negative oxygens. Now, this is different from an ionic molecule. Okay, with ionic compounds are made up of metals and nonmetals. Right? So if you look at the periodic table, you're looking for the metals on the left and then the nonmetals on the right. So an example of an ionic compound would be sodium chloride, which is our table salt. Okay, so sodium, Na, and then chlorine, Cl. Each of those, when they react together, the sodium is going to donate the electron to the chlorine, and the chlorine is going to receive from this sodium. You have two different ions, the metal and the nonmetal. The metal becomes positively charged and the nonmetal negatively charged. And then when dissolved in a solution like water, they become free ions, where each of those individual ions carries a full charge. So that's different here from the polar molecules. So for example, water carries a slight charge to it. Polar molecules are attracted to water, right? Whereas nonpolar molecules, for example, are fats, fatty acids. They're hydrophobic. And phobic, phobia, referring to sort of fear of. They're not actually afraid, but they repel water. Okay, same idea. All right, so your polar molecules are hydrophilic and nonpolar molecules are hydrophobic. So this diagram here is showing you slight polarity here to water, where you have the slightly negative oxygen and the slightly positive hydrogens. When these water molecules are in close approximation to one another, there's slight attractions, okay, between the hydrogen of one water molecule and the oxygen of another water molecule. And these slight attractions are known as hydrogen bonds. This is what gives water its unique property of having surface tension. Okay, so here you're seeing this example here of what we probably take for granted, but what is a water droplet? Okay, the water droplet, it forms this spherical shape because it's the most optimal shape at which it can reduce the surface tension in that form. So another way to look at this is, say you're at a body of water, like a pond or something, and you look at the surface and you can see little spiders that are skipping around, or little bugs that are on the surface there. The reason that they're able to stay afloat is because of the surface tension created by these hydrogen bonds. So at the surface of the water, there's a slight tension which, if not enough pressure is applied, allows those smaller insects to be able to live and stay afloat on top of the water. Now you can experience this surface tension. Say you were to go and do a belly flop from a very high level, say you were cliff jumping or something. If you increase your surface area and you go and you lay flat horizontally right on that water, that is going to hurt a lot, right? And that's because of that surface tension created by these hydrogen bonds. Okay, so next time you do that, I want you to hit the water and think, oh, those hydrogen bonds. All right, so water is a polar molecule, slightly positive end and a slightly negative end. The way that that interacts with ionic compounds is that this slightly positive and negative charges are attracted to the positive and negative cation and anions in ionic compounds. Okay, so here we're looking at sodium and chlorine. So this is an example of your sodium chloride. This is your salt. Remember I mentioned ionic compounds. The sodium is giving the electron, its outer electron, to the chlorine, and they are then bound together by those ionic bonds. What happens is the slightly positive, you can see it here, the slightly positive hydrogen is going to be attracted to the chlorine, and the slightly negative oxygen is going to be attracted to the sodium, the cation. And so what happens is when you put an ionic compound into water, it's going to dissolve readily. Now compare that, looking at an ionic compound, to what's known as an amphipathic molecule. Okay, so an amphipathic molecule has both, right? Like an amphibian can live both on land and in the water, right? Amphi means both. So an amphipathic molecule contains both polar regions and non-polar regions. So this is an example here of our phospholipid molecule. And the phospholipids make up our cell membrane. And the phospholipid is made up of a polar head with a phosphate group, you can see right here. And then it has two fatty acid tails. So the fatty acid tails, remember I mentioned before, fats are nonpolar, which means they are hydrophobic. All right, so the hydrophobic regions are going to orient themselves towards one another to stay away from the water. And the polar heads are going to orient themselves towards the inside and outside of the cell where water is in abundance. All right, we're going to get to look at this in a lot of more detail when we get into looking at this cell next week. Okay, so we're going to move on now to looking at macromolecules. So remember, we're at that basic level of organization, the chemical level, and we're looking at the largest structures. These organic macromolecules are formed by living organisms, and when we eat any food, we take it in, we are breaking down when we talk about breaking down into nutrients, a lot of that is the macromolecules that we are breaking down further into their individual monomers so that we can use them in different parts of the body. Now, all living things are based on carbon atoms. Okay, but there are 4 primary atoms that make up all living things. So when you think of an organic molecule, I want you to think of the following 4 primary atoms. So we have carbon, oxygen, hydrogen, and nitrogen, so COHN. Okay, carbon, oxygen, hydrogen, and nitrogen. Those are the 4 primary atoms that you're going to find making up all of our macromolecules. Now, there are 4 main types of macromolecules, as we talked about previously. 3 of them you find on the nutrition label. Okay? Proteins, carbs, and fats. Fats are your lipids. Okay, so proteins, carbohydrates, and lipids, those are all found on the nutrition label. The reason why you see them there is because they are providing us with caloric value. Okay, they are providing us with energy of some sort. The reason that nucleic acids are not included on the nutrition label is because they do not provide us with any caloric value. Okay, we can't obtain usable energy from breaking down DNA and RNA from animal and plant cell products. Okay, it's like anytime you eat, say, a burger or a salad, right? Everything, all tissue is made up of cells. And in every cell, you're going to have the DNA of that organism, right? So say you eat a salad, you are going to be taking in the nucleic acid from that plant. So although it doesn't offer us any caloric value, what we are able to do is to break down that DNA into its individual monomers, the nucleotides, and then we can use those nucleotides when we are creating our own new cells. So we use those as our own building blocks. Okay, so the 4 main macromolecules: proteins, nucleic acids such as DNA and RNA, carbohydrates, and there are lipids, which are fats. Nucleic acids don't provide us with any caloric value, but we still use the building blocks when we're creating our own new cells. So the key element here is that the macromolecules that we're talking about, those 4 main classes, they are made up of individual building blocks, and those building blocks are known as monomers. Okay, and we use those building blocks, we use monomers to build up a more complex polymer. Okay, so the monomer is the building block, and the polymer is the macromolecule. Okay, so another way to say it is our 4 main organic polymers, those would be the proteins, carbs, lipids, nucleic acids. Okay, so the polymer is the macromolecule, the monomer is the building block that makes up that molecule. So here we're looking at an example of our macromolecule protein. All right, this is a very simple protein that's been made up of 3 individual monomers. Okay, and those monomers are the amino acids. Okay, so you can see here's one amino acid here, linked to another by a peptide bond, linked to a third by another peptide bond. Okay, and so that's how we create sort of that initial primary structure of a protein. It's just simply amino acid linked with amino acid linked with amino acid. So it's very important as we're going forward, you need to make sure first and foremost that you know the 4 main macromolecules that we've talked about, and then you need to know what each of them is built from. Okay, so you need to know the monomers. Now, the way in which we put together these macromolecules, right? Because I showed you this here and I said, well, this amino acid's linked by this peptide bond, right? That's a covalent bond. Here you have another amino acid linked by another peptide bond to this amino acid, okay? And so here you've got 2 bonds. Well, at some point we had to put this together, right? We had to create this complex polymer structure. So to do this, we use 2 different types of reactions to build, one to build up and one to break down. So to build up a larger molecule, and it's in the name here, okay, we use a dehydration synthesis reaction. So there's 2 elements to this, this name that sort of tell you exactly what's going on. All right, so here's our example. Remember, synthesis refers to putting together, synthesizing. Okay, so we are in this case building up. So we're taking monomers. You can see here in this example, amino acid plus amino acid is going to give us our protein. Okay, dipeptide, that's just another name for a protein with 2 amino acids. So with a dehydration synthesis reaction, what ends up happening as we build up our polymer, we need to remove water. So from one amino acid, you remove this OH group, which is known as a hydroxyl group. And from the amino N from this amino acid, you remove the hydrogen, and that creates H, two H's, and an oxygen. So that's your H2O. So we take that H2O out, and you can see that illustrated here, okay, in the equation. Once we remove the hydroxyl and remove the hydrogen, now we need to stabilize and create a peptide linkage here between the carboxyl group from this end of the amino acid and the amino group here on this end of the amino acid. Okay, so when we remove water, and then that allows us to join the two amino acids together. So you can see that here, okay? And what we end up with is a polymer, the protein, plus a leftover water molecule. So what's happening here, when we're building up a larger polymer, and this is with everything, okay, not just with amino acids and proteins. Anytime you're taking the monomers and you're putting them together to make the polymer, you are using a dehydration synthesis reaction. Okay, so you are synthesizing a larger polymer, and to do so, you have to remove a water. Okay, so dehydration, removing water from your body, right? You're lacking water. So that comes into play here. Okay, so dehydration synthesis reactions, you're removing a water and you are assembling that protein. Now, the other reaction that we use, and this is to help us to break down polymers into individual monomers. Okay, and the reason that we do this, one example, the polymer, the complex carbohydrate glycogen, is how we store up extra glucose in our liver and in our muscles, so that if we're ever fasting or starving and we need glucose for energy, we can then break that glycogen molecule down and release individual glucose units. Okay, so that's just one example of why we would be breaking down a polymer into its component monomers. Okay, so hydrolysis is the reverse of dehydration synthesis. So with dehydration synthesis, we were building up a complex macromolecule. Well, now we've stored a macromolecule such as glycogen in our liver and muscles. Now we're fasting and I need some energy. So I'm going to start to break down that glycogen using hydrolysis. Okay, and again, look to the terms. Let's define this. So split it up, hydro referring to water, and then lysis refers to breaking down or breaking apart. So you're using water to break apart a molecule, right? So you're using water in this case. Water is now brought back into the equation to break the peptide bonds and then to bind the hydrogen back to the amino side and the hydroxyl group to the carboxylic acid. Okay, so water is brought into the equation here to help break those bonds, to release the individual monomers, and now we can use those monomers, specifically if we're looking at glucose here, we can use that glucose to then feed into glycolysis and Krebs cycle and produce energy for us. All right, so here's another example. Before we looked at amino acid 1 and 2, putting them together through a dehydration synthesis reaction. And now what we're going to do is we're going to break apart a complex protein and we're going to split those bonds. Okay, the peptide bond, we're going to break that using water. Okay, so water comes, you have the dipeptide plus water. That's going to then break up that peptide linkage, and now you end up with your two separate amino acids. So I can't emphasize this enough. You need to create as many charts to help you summarize the information that we're going through on a regular basis. Okay, so I try to add charts throughout, and sometimes I even, they're in the worksheets or posted in the learning module section. In this case here, just ensure that you've printed off this page, full page, and fill it out so that you can put this to memory. Okay, you need to know and be able to identify the individual monomers and which polymers they create. Okay, so we've looked at protein in quite a bit of detail here. That's been our example all throughout. So you know the individual monomers that make up proteins are your amino acids, and the polymer is the protein. Then as we're going through the rest of this lecture and next week's lecture, I want you to then take a look at some of the special identifying features of those macromolecules. Okay, so we're going to start here looking at the first of the four macromolecules. We're going to be focusing on proteins. So from previously in lecture, by now you understand that proteins are made up of individual amino acids. So the amino acids are the monomers and the protein is the polymer. So what type of reaction are we going to be using to take individual amino acids, the individual monomers, and putting them together, linking them up through those covalent bonds? Okay, that would be your dehydration synthesis, right? Where you're building up a larger molecule. So you're probably looking here and thinking, okay, I get this. So we have different types of amino acids. I can see they're all different colors here. And then we're linking them all up. Perfect. And then you jump here and you think, how do I go from this rainbow of color to a brown blob? Okay, so stick with me here. This is just showing you a small portion of what the entire polypeptide chain would be. So example, a complete protein like this, you're probably looking, at hundreds of amino acids, at least, if not thousands, that would be making up a more complex structure like this. So this portion of the polypeptide chain, you may just find it right in that region here. Okay, so the key here, we have one amino acid and another amino acid. And as I mentioned before, when you are putting them together, you're going to be linking them via a peptide bond. And that peptide bond is going to be formed through a dehydration synthesis reaction. So the hydrogen from the amino group, this is the amino, and the hydroxyl group here from the carboxylic acid side of the amino acid, they are both going to be removed from the molecules and then combined to form water and then removed from the equation. So there's your dehydration component. And then the peptide bond is going to be formed between the carbon and the nitrogen. Okay, so amino acids, those are the monomers, and they have a simple basic structure. And where they are different from one another comes down to the variable side chain here, which is identified just by that letter R. So each amino acid is going to contain a carbon atom to which we have the three following structures bound to it. Okay, so we have an amino group, which is your NH2. We have a carboxyl group, COOH, and then some sort of functional group. And this is what identifies one amino acid from the other. We have 20 different types of amino acids. 9 of them are essential. Okay? And what differentiates one from the other, you can see here, is all the base structures remain the same, right? We have that alpha carbon, we have the amino group, and then we have the carboxyl group. It's the variable side chain, that functional group that's identified or highlighted in blue. Okay, that's what's differentiating one amino acid from the other. Now, 9 of these are known as essential. So here's the definition for you. Essential amino acids are those that we cannot produce ourselves and we require from our diet. So the 9 that we have here listed: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. What you need to know are that we have 20 different types of amino acids, and the different types based on the variable side chain. Okay, and 9 of those 20 are essential, and those essential ones we need to receive from the food that we eat. Amino acids are the base structure for non-steroidal hormones. So one example there, we use tryptophan, which I'm sure you've all heard of before. It's the amino acid that you get from eating turkey, a number of other places as well. Tryptophan is an essential amino acid, and we use tryptophan as sort of the base building block to make the hormone serotonin. So that's your good mood hormone. Tyrosine is another amino acid that we use to make dopamine, which helps you focus. Epinephrine, norepinephrine, those are your stress hormones that we'll talk about when we get to the sympathetic nervous system. And then your thyroid hormones as well. Now, you might be looking at this table thinking, okay, that is a ton of information. How am I going to pare this down for myself? The main focus at this point in our curriculum is to understand that there are different types of proteins. Okay, so understand what the different types are, and then generally what the functions of those types are. Okay, the examples that are all listed here, we have an opportunity. We are going to be talking about every single one of these, I'm pretty sure, as we move throughout the rest of both biology and anatomy. Okay, so you're going to see them in different capacities as we move throughout the rest of the course. So different types of proteins. So starting off here, we have enzymes. So enzymes are functional proteins, and they're used to help to catalyze reactions. So the condensation or dehydration synthesis type reaction that we talked about, there are enzymes that can actually help facilitate that reaction. So they can help monomer 1 and monomer 2 come together and bind to form the more complex polymer. There's also enzymes that help to break down. So we're going to be looking at a lot of these enzymes throughout the course. Lipase, one example. Other digestive enzymes we have. Amylase breaks down our carbohydrates. Nucleases break down nucleic acids. And proteases are the general class that break down proteins. Anytime you see ASE, I want you to think enzyme. All right, and we're going to talk about lactose intolerance as well when we look at carbohydrates next week. And you'll see that lactase is the enzyme that breaks down lactose. Okay, so lipase is the enzyme here that breaks down our lipids. All right, so ASE is typically referring to an enzyme. We also have proteins that are involved in protecting the body from either pathogens or other damage. Antibodies, I'm sure you've all heard of antibodies, especially with all the talk about vaccination in relation to COVID-19. Antibodies are proteins that are produced to be able to target specific pathogens and help to flag them or remove them from the body. Membrane proteins. So within our cell membranes, we can have proteins that are embedded there for a number of different purposes. Some of them act as active pumps, like the sodium-potassium pump. We're going to look at that in a lot of detail when we get to the nervous system to see how the neurons are able to actively pump sodium out of the cell and bring potassium into the cell, creating a concentration gradient that they can harness to be able to produce a nerve impulse. Now, sodium-potassium pumps exist to create that gradient within all of our cells. So kind of a general rule is sodium is always higher outside the cell and potassium is always higher inside the cell. Now, we also have channels located within our cell membranes as well that can allow for the movement of specific ions from the external environment, so the extracellular, to the intracellular environment. We also have proteins on the surface of our cells, such as antigens. I'm sure you've heard that term maybe before. Antigens are proteins that are used, think of them as like flags or markers on the surfaces of all of our cells. There are self-antigens, which allow our immune system to determine, hey, that cell should be here. And then we have, there are foreign antigens that our immune system or immune cells are able to detect and determine, hey, like that's not supposed to be here. We need to get rid of it, whether we destroy it or I go tell somebody and they come and destroy it. We'll get to talk about the immune system a lot once we get to next semester. Also, antigens on the surfaces of our red blood cells identify what blood type we are. So we have two different types of antigens. You have your ABO type of antigen, and then also the rhesus factor, which is identifying whether you're positive or negative. There are contractile proteins that are involved in muscle contraction. So muscular system, we're going to be looking at actin and myosin and how they work together to help to form a muscle contraction. Specific transporters as well. Hemoglobin's a great example of this. Hemoglobin's a globular protein made up of 4 different protein subunits. And each of those subunits is capable of transporting an oxygen molecule. So hemoglobin is the molecule, the protein molecule, that's responsible for transporting oxygen to our tissues from our lungs. We also have structural proteins. We've talked about collagen, right? When we looked at connective tissue, collagen was that protein fiber that's responsible for producing strength and durability. As well with actin, it's a protein that forms intracellular microfilaments that allow for cells to move. And last but not least, regulatory proteins. We're going to look at, first off here, you have insulin, growth hormone, just some examples of protein-based hormones. So those are your non-steroidal hormones that are involved in communicating throughout the body. That's pretty much the main role of a hormone. And troponin and tropomyosin, 2 regulatory proteins that help to regulate how actin and myosin, those functional proteins, are going to be performing a muscle contraction. So the main, I've given you the examples here and we talked about that, but the main thing to focus on is going to be the main types. So those classifications, and then just generally what that function is that they're going to be. So a protein structure is complex, and it's the order of the amino acids. So this is a very important point here. Okay, so we saw that at the very beginning that we had a certain order of those 5 amino acids that we linked together. Okay, when you consider that you have hundreds of amino acids, thousands of amino acids that are going to be in sequence, that sequence needs to be exactly a specific way for that amino acid to be able to fold a certain way. And the shape of a protein is specific to its function. All right, so let's take a look here at how we go from just a simple polypeptide chain to creating a more complex globular structure. So first off, remember we took those individual amino acids. How many of them do we have? 20, right? 9 are essential, 11 non-essential. Out of the 20, we are going to be putting together a protein using a dehydration synthesis reaction. Okay, so we're linking up, creating peptide linkages between each of these amino acids. Now we've got an entire sequence, a polypeptide sequence here. Okay, so that initial polypeptide chain, that's known as your primary structure. So then based upon regions that have certain sequences, that chain is then going to naturally fold into a secondary structure. So there's certain portions that are going to form, you can see here the green, it's going to form beta-pleated sheets. Essentially just think of them as folds, okay? And then other portions of that polypeptide chain are going to form alpha helices. Okay, so that's that helical structure. We see a double helix in DNA. The alpha helix is just that single strand of amino acids is going to be sort of forming a helical structure like a coil. So then that's the secondary structure. The tertiary structure is formed based upon certain regions of that polypeptide that are hydrophobic and other regions that are hydrophilic. So picture this. You're going to have certain parts that are going to be hydrophobic, and they're going to want to fold towards the interior of that protein because they want to stay away from the water. And then you're going to have others that are hydrophilic, and they're going to be located more towards the outside or the periphery of that protein. All of the sequence is all going to lead to creating a globular protein that's going to have a specific structure based upon a number of different interactions within that molecule. Understand the difference between your primary, secondary, tertiary, quaternary structure. Here with the quaternary structure, you also have the interactions between different parts of the protein that are playing a role in creating different weaker linkages, as well as hydrophobic interactions that are going to keep those portions folded towards the interior of the protein. So as I mentioned before, the shape of the protein determines its function. Now, there's certain conditions proteins can become denatured. So here's our protein, right, that we've created. Okay, this is our globular protein. With this specific shape, it has a very specific function. Now, two elements, temperature and pH, can alter the way that hydrophobic interactions occur with one another and can actually denature that protein. So the different interactions between parts of the chain that were keeping this in this structure, they can be interrupted by seeing increases in temperature or increased acidic conditions. All right, so once this happens, the proteins are no longer in their active form, they now become inactive. Now, proteins are processed in the cell and modified in some way. Some proteins can be combined with carbohydrates to form glycoproteins, and others can be combined with fats to form lipoproteins. So just different modifications. The modifications occur within the Golgi apparatus, and that's a structure that's responsible for modifying and for the packaging our proteins before they go to do whatever it is that they're destined to do. So with a glycoprotein, you'd simply be adding a carbohydrate or attaching that to the protein. And with a lipoprotein, you'd be adding lipids or fats. Now, as we mentioned before, enzymes are the functional proteins that are going to have a specific shape that is going to allow them to fit precisely with some other molecule. Okay, now enzymes cause the chemical or molecule that they fit with to undergo a reaction. So remember, by definition, enzymes are catalyzing reactions. They are helping the reaction to take place. So here's an example of our enzyme. Let's say that this is our lactase enzyme. So that enzyme is going to have specific regions where lactose is able to bind with it, okay? And that's known as the active site. So when the lactose molecule, which is a disaccharide, when it binds with the enzyme lactase, lactase is able to then catalyze that hydrolysis reaction that's required to then break apart the molecule. Okay, so enzymes are able to catalyze, which means to enhance, speed up, or facilitate those reactions to take place. So here we're looking at an example of taking a disaccharide, okay, or any polymer made up of two monomers. The enzyme then it's going to bind to that molecule, and then it's going to break the bonds through a hydrolysis reaction, and now we'll end up with individual monomers. So here's an example of our lactase enzyme and how it works specifically with lactose. So lactose is a disaccharide, and then through a hydrolysis reaction, right, we're putting water in, and that's going to help to cleave the bonds of lactose into galactose and glucose, the individual monosaccharides that make it up. And so if an individual has sufficient amounts of lactase enzyme within their small intestine, then they're capable of being able to process lactose from dairy products. Okay, lactose is a sugar found in milk. Now, in individuals that are lactose intolerant, the reason that they're intolerant is because they lack this specific enzyme. All right, and so because they lack the enzyme, lactose stays in its disaccharide form, and so it remains in the small intestines. Now, the bacteria in our intestines are then able to feed off of this, because remember, it's a sugar. And when they feed off of it, they produce lots of gas, and that's what causes the bloating associated with lactose intolerance. Okay, so moving on from proteins, we're now going to focus on nucleic acids. We have two types of nucleic acids, DNA, which is deoxyribonucleic acid, and RNA, which is ribonucleic acid. Each DNA macromolecule contains hundreds to thousands of individual genes, and genes are specific regions which act as the blueprint for a specific RNA molecule called messenger RNA that's going to code to make a specific protein. Okay, we're going to fill in all the blanks after Christmas in the winter term when we spend the first 3 weeks looking at DNA. We're going to be looking at replication, transcription, and translation. So DNA and RNA consist of monomers called nucleotides, and each nucleotide, so this is our diagram of a nucleotide with a phosphate attached to the sugar attached to the nitrogenous base. Okay, and that you can see outlined here. So you have your phosphate group, you have the sugar. Now this can be a ribose sugar or a deoxyribose sugar. In this case, we're looking at a deoxyribose sugar, so we know this nucleotide must be for a DNA molecule, okay, because D in DNA stands for deoxyribo. And then attached to that sugar, to the first prime carbon, we have our nitrogenous base. Now, there are 5 different types of nitrogenous bases, okay? In DNA and RNA, we have cytosine, guanine, and adenine. Okay, the difference is between DNA, where we have thymine, and RNA, we have uracil. And there's only 2 possible base pairs. So here we're looking at DNA, double helical structure, so there's 2 strands, and you're always going to have the one strand is going to pair with a complementary base pair on the opposite strand. And so these base pairs are always going to hold true. Adenine is always going to pair with thymine, or thymine with adenine, okay? So A and T go together, and cytosine is always going to pair with guanine, so C and G go together. Now, holding these complementary base pairs together are hydrogen bonds, and you'll notice adenine and thymine contain 2 hydrogen bonds, and guanine and cytosine have 3 hydrogen bonds that hold them together. Then linking up the sugar phosphate, you create what's known as a sugar phosphate backbone of our DNA molecule. And that consists of phosphodiester bonds. Now I have a diagram that's included in the online learning module section. So take a look at that. That's going to give you a perspective as to the individual bonds. So here's the hydrogen bonds right there, okay, holding each complementary molecule together. Remember, hydrogen bonds are relatively weak, which is great because we can then unzip the DNA molecule to then go through the process of transcription. The phosphodiester bonds are what are linking the phosphate group of one to the sugar of another. And then the sugar, you can see here, here's all of those pentose sugars. Because we're looking at a double-stranded DNA molecule, you know that's a deoxyribose sugar. The sugar is then bound to the nitrogenous base by a glycosidic bond. Okay, so just from this one diagram, you've got 3 different bonds, okay? So holding the 2 complementary strands together, we have the hydrogen bonds. Okay, so holding the 2 complementary base pairs together, we have the hydrogen bonds. Between the sugar of one nucleotide and the phosphate of another, we have the phosphodiester bonds. And then between the pentose sugar, so the deoxyribose sugar, and the nitrogenous base, we have the glycosidic bonds. All right, so taking a look at a sequence of DNA here, ACTTT, CCTAG. Okay, we know what all of those letters stand for, right? This is not gibberish. You know that each of those codes for a specific nucleotide, right? A nitrogenous base that's on that nucleotide. So A, adenine, C, cytosine, T, thymine, G, guanine. So we have those 4 different nitrogenous bases, and they all are going to bind on the opposite strand to their complementary base pair. Okay, so let's think back. Let's go back. Okay, adenine's always going to bind with thymine, and cytosine's always going to bind with guanine. Now, you have your sequence of DNA. So the opposite strand is going to read TGAAA, CTA, GGATC. Okay, so the A is always going to bind with the T, and the C is always going to bind with the G, and vice versa. All right, so that's how you could come up with a complementary strand based on your initial sequence of DNA for me. All right, so be able to do that. And now, if I was to say that this was the strand of DNA that we were going to be taking a working copy of, what would be the RNA sequence? So here in this case, remember, instead of thymine, RNA contains uracil. So here we would have a U that's going to bind with the A. Okay, so let's work through this. So binding with A, you'd have the U, G. Now T is going to always bind with A. Remember, we have the adenine, but it's the uracil. So instead of having any thymines in this RNA sequence, we're going to have uracils replacing the thymines. So it ends up UGAAA, CUA, GGAUC. All right, so just to compare, DNA is a double helical structure, right? Double-stranded, whereas RNA is single-stranded. The nucleotides in DNA, adenine, thymine, cytosine, and guanine, whereas uracil replaces thymine in the RNA molecule. The sugar is a deoxyribose in DNA, and in RNA is a ribose sugar. DNA is located and housed in the nucleus only. Okay, so RNA is transcribed in the nucleus, but we have these nuclear pores that allow the messenger RNA to then move into this cytoplasm, right? That's the jelly-like suspension medium inside of our cells, okay? And then that's where the protein is going to be made from that messenger RNA. The function, DNA acts as the store of our genetic information, and RNA is used as a template. That's key, okay? So RNA is like a working copy of our DNA. So the DNA is stored inside the nucleus. The RNA is a working copy that leaves the nucleus and goes off into the cytoplasm to then have proteins produced from it. Okay, and we're going to look at this in a lot more detail as well when we get to looking at the cells. We're going to talk about these different structures and how this occurs as well there. All right, so that brings us to the end of this week's module. So I look forward to seeing you all in our tutorial this week. Please bring your questions, but if you have any questions in the meantime, before I see you, please feel free to send me an email and I'll get back to you as soon as possible. Otherwise, take care.